Hi everyone, welcome to Quantum Photonics. Let's wait for a while until more people come, and I'm trying to ping our guest, Dr. Dimitris Pinotsis.
Okay, Cecile, the mic is yours. Thank you.
Thank you so much, Darcel. Welcome everyone, and welcome to Quantum Photonics. Hi, Shera and Wissam, and hi to our guest, Dimitris. So, welcome to Quantum Photonics.
Hi, Cecile and everyone. Thanks for having me. It's a pleasure to be here. Can you hear me okay?
Yes, I can hear you well. So, thank you for coming. I will be introducing you shortly. Um, so, we will wait for maybe 30 seconds for more people to come in. As there will be a playback for this, I would like to start introducing our special guest for today, who has a very interesting work about the electric fields and the brain. Dr. Dimitris Pinotsis is a theoretical neuroscientist with a PhD in mathematics and a Master in Science in Theoretical Physics from the University of Cambridge in England, where he worked with Onassis Foundation. And after graduating, he published more than 15 papers in mathematics and physics journals, and then decided to pursue a career in neuroscience, his true passion. Dimitris worked with Peter Greenrod and then spent 6.5 years at the University College London working with Carl Friston in machine learning and developing mathematical methods for the analysis of brain data, then continued at the Massachusetts Institute of Technology, MIT, where he worked with Earl Miller on using predictive coding and deep neural activities to address fundamental questions in cognitive neuroscience. In 2018, he joined the Center for Mathematical Neuroscience and Psychology at City University of London as an assistant professor lecturer. He is now an associate professor reader at the same university and a research affiliate at MIT's Brain and Cognitive Science Department. Dimitris has received over 10 fellowships from Cambridge Isaac Newton Trust, Onassis Foundation, NATO, Bernstein Organization, and others. He is the recipient of several awards, including OCNS Award, a Smith-Rayleigh Prize at the University of Cambridge, and a Poincare Institute Award. Dimitris' research has been funded by the U.S. Air Force of Scientific Research, Picower Innovation Fund, UKRI, Innovate UK, Gambling Commission, EPSRC, and the Wellcome Trust. It spans diverse areas, including machine learning, the analysis of big data in neuro-imaging, theoretical neurobiology, and nonlinear systems in mathematical physics. So guys, I present to you Dr. Dimitris Pinotsis. Mic to you, Dimitris.
Thanks so much, Cecile, for this wonderful introduction. I'm not sure I deserve it. So I hope everyone, or at least those members of the audience who can do that, have my slides open, the PDF file. And the first page, there is a brain, and the title that I have put for this registration is "Where in the Brain Can I Find My Thoughts?" So hopefully everybody can see that. So what I will do is I will be referring to different slides as we go along by their numbers. So slide 1 is this brain with the thought, basically. So hopefully everybody is on the same page. So indeed, I want to talk about electric fields generated by our thoughts and our memories in particular, which one can think is the most elementary kind or sort of one's thought. And not only basically the electric field generated, but the neurons that generate these fields and help us maintain our memories and construct and process our thoughts. And the bigger question here, of course, is, I'm going now to slide 2, is information processing in the brain. How does this happen? That's the thing that motivates, let's say, this line of work and the large body of work by myself and others in cognitive neuroscience. Here is a bit of a historical summary, if you will. On the top left of slide 2, you see this image with a Pavlovian dog, for example, or rats. So the beginning of cognitive psychology and the study of information processing in the brain goes back to behaviorism and the study of reflexes, basically. So the assumption back then was that behavior is, of course, a result of information processing in the brain, and really one major output of this processing is reflexes and positive or negative reward signals. It could be reinforcement signals or punishment signals. And again, this is, of course, an old story in cognitive psychology. Then on the top right, I have a schematic of an initial neural network from the 50s or 60s. So an obvious weakness of behaviorism is that, of course, did not give us much insight in terms of how information processing and behavior happens inside the brain. So connectionism came about trying to answer that and had some initial toy models, the first neural networks by, for example, people like Rumelhart, and the famous work by McClelland regarding the past tense of English verbs, etc. So after connectionism, of course, which offered the first models of information processing in the brain, we had in the early 90s, the revolution of neuroimaging. Things like PET and fMRI, and later, of course, electrophysiology. So moving now to the bottom left image, what neuroimaging tried to do is to find brain signals and correlate them with behavior. Basically, so brain activity correlating with putative mental states, and that was the basis, of course, of cognitive neuroscience and cognitive psychology. And if you will, the recent deep neural network revolution, at least in the field of cognitive neuroscience, and the schematic I have on the bottom right side of this slide, these deep nets, is a follow-up of that line of work, and combined, of course, with connexionism. So we have high-resolution brain data from neuroimaging, and now we have to build these very powerful neural networks, deep neural networks, that allow us to really mimic and predict these very complicated brain responses. And they have so many parameters, but of course they also have some weaknesses. A very one commonly mentioned for deep neural networks is basically that they need, for example, supervised training using very, very large data sets. And this does not resemble at all the amount of data they need, and the process that we use for teaching these networks does not resemble how, for example, babies learn to do simple things. So that's a historical review of, or a few things, a few remarks, introductory remarks for information processing. And I'm moving now to slide 3, which is the focus now of this presentation, of this talk. So slide 3 now we talk about these groups of neurons that represent or maintain information in the brain, and we call them neural ensembles. Another name you might have heard is the one also used by Head, so cell assemblies. So neural ensembles and cell assemblies are these neurons that maintain and sometimes process stimulus information. So let's see a few examples. Again, slide 3 now, top left, you see a rat under an optical microscope, and you also see some green dots in the green cloud. I hope everybody can follow that. So what are these green dots and the green cloud? These are the neurons in the rat visual cortex that are activated real time when the rat is looking at the images. So for each different image that we show to the rat, we have a microscope on its head that you see here on this image, and then we record that different neurons are activated. And what could the image be? So the image could be something like, it could be a natural, an image, or it could be something now, if you go to the right-hand side of the top row, you see again similar clouds of neurons and white dots, in this case, again with optical imaging. And there are 9 small squares, and at the top left corner of each of these squares, we see some small colored bars. So these are the stimuli that we show, for example, to the rat, and they're bars of different orientations. So you see, for example, that the very top left square, we have this yellow bar oriented, for example, at 60 degrees, if you like, and then the next to it, the red one, is at 0 degrees, etc. So for each orientation that we show to the rat, we can see, look, which neurons are activated in his brain, and we see these small dots. These neurons are activated at the same time, so they are synchronous. They have synchronous spiking, and these neurons are what we call neural ensembles or cell assemblies. And now at the bottom row, right-hand side, you see an fMRI image. So here we have a part of the posterior parietal cortex, another area in the brain, and you see this color, different colors that range between red to purple for different numerosities. So we show to a monkey in this case, stimuli like dots of different numbers. So let's say 1 dot, 2 dots if it's yellow, 3 dots if it's green, and so on till 7 dots for the purple. And you see different parts of this of the monkey's brain respond. So again, we have a map, basically a tomographic map of here numerosity preference, and the corresponding parts of the brain that respond to different numerosity levels are neural ensembles. So these are the neurons that are selective to this particular value of the stimulus. And the last plot we have on this slide at the bottom row, on the left-hand side, is work by Earl Miller in his lab and Tim Bushman. And I will be talking about our work with Earl throughout this presentation. So in that earlier experiment in Earl Miller's lab that you see here, the monkey had to basically decide and remember some stimulus. It doesn't matter what it is. It could be, a random dot, a cloud of random dots. But the crucial thing is what you see here on this schematic is on the right column. There are in a sense two columns here. On the right column, you have a grid. You have these small dots on the monkey brain around the PS area, the sulcus. And basically, you see these blue lines. There are some straight lines that connect dots. So the dots are electrodes, an electrode array that covers the monkey brain. And then the blue lines on the top and the red lines, straight lines on the bottom, correspond to pairwise correlations between two electrodes. So basically, this is another way to visualize synchronicity between different parts of the brain. So you can think that the blue lines tell us which neurons fire synchronously in the blue condition, and the same thing for the red. But what is important here? The important thing here is that these conditions are not like orientation, let's say values, like in the previous example, or numerosity values of the stimulus. These are more abstract. So here is rules, basically. So when the monkey has to take a decision based on rule A, we see the blue pairwise correlations. And then on rule B, if the monkey uses rule B, we see the red. So what is the crucial point I want to hopefully explain here? That these ensembles, this group of neurons that are activated, and they represent some part of information in the outside world, inside the subject's brain, does not only relate to stimulus properties like orientation or numerosity or something else, but it could also relate to abstract notions like rules in this case. So that's a little bit of overview about neural ensembles. The next slide 4 tells us now when could we expect to find these ensembles, these groups of neurons in our brains, both humans and animals. So the neural ensembles mean, like we said already, synchronous firing and oscillatory responses. So some kind of periodic activity that repeats over time and leads to this coordinated firing across many neurons or many parts of the brain, like we saw in the earlier slide. And this sort of oscillatory response is a rhythmic, coordinated, synchronous activity, underlies cognitive functions, like on the first line here, different perceptual paradigms, attention, memory, executive control tasks. And what is the point? The point is that to perform this kind of cognitive tasks, like a memory task that will focus here, but also, like I said, attention tasks, we see similar things. We have highly parallel processing by the brain. What do you mean by parallel? I mean also spatially distributed, but also temporally bound information. So as we saw earlier, we have, for a certain stimulus, different neurons activated, and these neurons are in different parts of the brain, so there is some kind of spatial distributed processing, but also they're synchronous. So there is also temporal binding. So the neuronal discharges between different neurons in different parts of the brain are synchronized, as we said. And the hypothesis is that this synchronization of neuronal discharges binds information together into these cell assemblies. And this is the way that the brain selects behaviorally relevant information. So this is the way, this binding over time, this goes back to theories by Wolf-Singer and others, this binding hypothesis. So information integration relies on this temporal binding and also coherent representation of states in different neurons that are distributed in different parts of the brain. That's the hypothesis here. So oscillations, temporal binding, and spatial integration. So let's move to slide 5 now. Slide 5, where I show basically another illustration of this kind of idea of oscillatory, coherent, spatially distributed responses from now human data, interestingly, non-invasive. So you don't need to go to the microstructure necessarily to see all these things. In animals with invasive recordings and put electrodes in the brain, you can even pick up some of these responses even with common tools like magnetoencephalography or electroencephalography that we use for humans. So slide 5 again, what we see on the left column is something called Mooney faces, maybe familiar to some of you, some classical experiments by Rodriguez and al. 1999. So left column here you have on the top row a face, and then the bottom row again in the left column, this black and white image, you have an inverted face. So what this scientists did is they showed basically both the upright face and also the inverted image to human subjects, and then they recorded non-invasive electrophysiology data. And these recordings we see now in the right-hand side column, and there are two columns there, these reddish orange kind of brains that you see. There is a no perception column and the perception column. So the no perception corresponds to the inverted image. So when the subject does not perceive the face, while the perception column corresponds obviously to the normal image where you saw the face in the upside down, the normal, let's say, direction. Now you have 1, 2, 3, 4 rows, right? 4 different snapshots of brain activity. So you have the brain here, you have some small dots, green dots that are the electrodes on the brain. And then there are lines. There are two sorts of lines here. You can see green lines and black lines. Black lines are similar to what we saw in the earlier slides. They are pairwise correlations between different electrodes on the human skull, on the head. While green lines are basically desynchronization between these areas. So you see now, if you focus on the rightmost column, the perception column, and you start from the top, from 0 to 180 milliseconds, I'm reading the legend on the rightmost side of this figure, so different snapshots for different epochs in the experiment. We start from the top, going down to later epochs. So the first, there is no synchronicity basically, or no desynchronization, no black or green lines. But then if you go to the second snapshot, on the rightmost column from the top, you have 80, 100 to 360 milliseconds after presenting the face to the subject. And you see these black lines appearing. So these are pairwise correlations between different parts of the brain. In other words, this means that we have synchronous activity, oscillations, associated with the perception of the face. Now, if you look at the other column, the no perception column, you see the green lines, which means there is desynchronization. So, to make the long story short, what happens here is when we have conscious awareness of a face, we have synchronous activity. When we perceive the face, different parts of the brain synchronize to represent this information of the face in our brains. That's the simple one-line summary of this idea. So you have an active coupling of different neurons that are brought together and are hooked up in a network that produces oscillations, and by doing so, represents phase information and allows the brain to process this information. So hopefully the idea by now is to some extent clearer. So this is what we're interested in, right? There is a thought of the phase here, and we know where it lies in the brain. We can even access it non-invasively to some extent. So now moving to slide 6, where I summarize basically, and I give reference to some papers, and we have reviewed the literature in the paper that you see at the very bottom, our 2019 brain topography paper. So this kind of oscillatory responses are associated with maintenance and processing of stimulus information in memory tasks, in attention tasks. They are thought to underline the binding of input to cortical representations. So as I say in the fourth point here, again, slide 6. You can use that to describe information propagation in the brain, and also as markers to distinguish using oscillatory features like peak amplitude or frequency. So distinguish between healthy, let's put it like that, or information processing in controls versus information processing that takes place in individuals with some psychiatric disease or disorder, autism. So maybe, Cecile, shall we have the questions up later, I guess, or feel free to interrupt me if there is something. I'm not monitoring the chat here, if there is something else. Yeah, okay,
We shall have the questions later, I think.
Okay, great, no problem. So this is my introduction, basically, so far. And this is what we'll be talking about. And these neurons will generate some electric fields, like I said. And that's what we'll try to talk. So slide 7 now. So we'll talk about the connectivity and how these networks are formed in the brain. Slide 7. And also the computation that might underlie and take place in these networks, and the semantics, basically, that are represented and processed. So this is what I would like to discuss in the remaining part of this presentation. And we have used a variety, as you will see, of mathematical tools like graph theory, dynamical systems, and also tools from computer science like deep neural networks and representation similarity analysis, LSTMs, autoencoders. So whatever is necessary to really characterize, on the one hand, the structure of these ensembles and their biophysics, and on the other hand, the computation that they perform. So let's start with the structure and the biophysics of these ensembles that will take us directly to the electric fields they generate, because these are bioelectric fields. So we need to start with the biological properties of these brain networks. So moving now to slide 8, that says structure of neural ensembles, and has the same images, more or less, the same pictures that I talked about earlier. There is one small point that I have put on the top of this slide that I would like to come across, which is that basically what we talk here is similar, of course, to other approaches that exist out there for studying and identifying neural ensembles. And there is a small twist. So what are these, first of all, what are the classical approaches that people use to identify, find where in the brain these neural ensembles are? So traditionally we have two sorts of approaches, roughly. We have spike sorting and clustering approaches that are based on pairwise correlations, similar to the straight lines we saw before in a couple of paradigms, in the Mooney phases and in the rules paradigm. So one thing you can do is measure activity spikes from a variety of neurons and then try to cluster them using k-means or something else. That's one thing. The other approach that people often use is some kind of dimensionality reduction, some kind of PCA, principal components analysis approach, where you get a latent space. And this is very similar to what we do. So this is the second approach, is what we take here, and I'll come to that. But there's a twist, like I said, and the twist is that we put something on top of that. So basically, we first evaluate the latent space using this PCA kind of approach, and then we map the latent space to the cortical path. So we give some biological description to this latent space by using inverse problem theory. So moving now to slide 8, which has this roadmap of our approach and some new theory that I've developed for this. So our starting point, so slide 9 now, slide 9. So we start with a neural network, which could be a Wilson-Cowan neural network, something very simple and very well used. And our first step is basically to reformulate, do some mathematical tricks here and analytical calculations to write it as a deep neural field network, so as a biophysical neural network, essentially. That can also be cast as something called a Gaussian linear model. So you see step 1 at the top of my roadmap here. So we go from a neural network to a Gaussian linear model that we can then train in step 2 using a variational autoencoder to get the latent states, something we will call the connectivity components, similar to PCA. And we'll come back to all this later. I'm giving you now the high-level description. Some things will not be clear, obviously, because we need to go into details. So once we have now the latent states, but this is again similar to people who know dimensionality reduction, it's the same thing. What we can do is step 3, we can reconstruct, based on the connectivity kernels, the weight matrix of our neural network, the biophysical neural network, and this will lead us to electric fields in step 4 if we put a dipole classical electromagnetism model on top, as we will see. This will give us predictions for electric fields, or if we use graph theory, it will allow us to characterize the topography or the topology of neural ensembles. And we have done this work. The topography and topology is the older work in the 2017 neuro-image paper. The recent paper that Cecile, maybe, and other people might have seen, and it's maybe in the summary of this presentation, it's basically the electric field work is the recent paper in Neuroimage 2021 or 22. And there is some related work. All this is about the structure of neural ensembles and the biophysics. There is also some related work about the computation, which is another paper from 2019. We'll see how much we can cover. I'm trying to be as slow as I can, and the main point is to really try and explain a few things. So let's dig a bit more into the biophysics and the structure of neural ensembles and try to see where the electric field comes in. Slide 10 now. So the first step we want to have is a neural field model, a biophysical neural network model of the ensemble. So what you see on the top left here is basically a square with blobs, these small circles of different colors. And then you have some numbers on this on the edges of the square, so you can think of this as a small cortical patch of a few millimeters, and the numbers you see are the millimeters, for example, that you are measuring activity from. What are the blobs, the different colors? So this is the LFP data you are measuring, the local field potential data, the depolarization, the electrical activity. And different colors here mean different levels of voltages, effectively. Some places have more voltage, some have lower voltage, basically. So you have a biophysical neural network, and this is really the way we describe the neural ensemble. And like I said, we use this neural field modeling, which describes spatially extended neuronal processes of a few millimeters on the cortex, my second point here. And then the third point, you don't need to worry about the math. These are known, of course, in the field of computational neuroscience, and they are used because they can explain the relation between spatial or biophysical properties of neural ensembles like conduction delays, dispersion of axonal connections, and local field potentials or even spike data. So observed brain dynamics. So this is the kind of models we use, and other people have used them, but there is a twist here that basically we have a way of finding the connectivity between different neurons in a neural ensemble from data. So we essentially what we do, I think it should be, actually it's on slide 12 now. Let's move on slide 12 if you don't mind, then I'll come back to slide 11. So slide 12, you see basically what we can do. We take this biophysical neural network model, we train it with local field potential, LFP data, as an autoencoder, and then this gives us the weight on the weight matrix, the connectivity matrix on the cortical paths. And this is what you see, slide 12. There are some panels here on the top left side, and if you read the legend, the vertical one says sending population or electrodes, and the horizontal, again, top left panels, we have the receiving subpopulation or electrodes, and then we have 6 panels. So basically for different memories. So let's go back to slide 11 now and see what this weight matrix corresponds to. So slide 11 has the task and the experimental setup. So what is the task? Very simple spatial delayed response task from Errol Miller's lab at MIT. And you see a schematic of the task here. You have a monkey, the monkey fixates, then there is a dot that appears on the visual field. There's a cue, as we call it. The cue disappears, so there is a delay period. And then the monkey has to very simply saccade to the presented location. So during the delay period that we're really interested, there are some neural ensembles, some groups of neurons in the monkey brain that maintain the angle. So where was the cue presented on the visual field? And then you see also on the top left side here, there were 2 monkeys, and we recorded data with these multi-electrode arrays, these small squares with the dots inside from 3 different areas, SCF, FEF, and PFC. Here we'll talk mostly about FEF, frontal eye fields. And I'll come to that in a minute. So you see also the size of these multi-electrode arrays. These are 2 millimeters long. If you look, again, monkey C, there is a small arrow near the multi-electrode array, top left of this top left panel. It says 2 millimeters, and there are 2 arrows, orange arrows. And then the separation between 2 electrodes on this array, it's about 400 micrometers. So we put these multi-electrode arrays on the monkey brain. We have several electrodes arranged in a square. They're very, very close to each other. And you see, if you remember, this is similar now. This square is similar to the square we had basically for a neural field model. And now what we do is we give numbers to these electrodes. So there's electrode 1, 2, 3, starting, for example, from the top left side of the multi-electrode array. You can say, okay, this is a very simple task. Why do I need to care about this? This is actually very useful in brain-computer interfaces because this is, say, the prototypical task or a very fundamental task that people use to see how they can use saccades as a means of communication. So you see here the monkey has to saccade to the remembered location, so they will train a neural decoder. So the task itself has to include some saccade. So what is the easiest thing to use the saccade for? Special location. So you teach the monkey to really saccade to whatever they were presented with. So that's the task. It's called, again, spatial delayed response. The monkey needs to respond based on the spatial location of the cue after some delay. And basically you have now, we go back to slide 12. Let's look again at this weight matrix on the top left. So you can think that you have this array, it measures activity from certain neurons, and then these neurons of this array are connected with some weights. And what you see here is some way of mathematically depicting these weights for different ensembles or different values of the angle that is remembered. So if you see, it says theta equals 0 on the top left panel, and then below that you have theta, the angle, equals 180 degrees, and so on. So we have, for different memories that are remembered, different acute locations, we have different weight matrices that we obtain. Again, by training our deep neural field model as an autoencoder using this monkey data that we recorded in this task from the frontal eye fields. And what is the interesting result here? The interesting result is that we find this band structure, this, let's say, translational invariant structure along the diagonal. So if you look at these weight matrices, you will see I have some either more circles or ellipsoids, blue dashed blue circles or red ellipsoids. To say what? To say that the way connection weights change with distance as I go away from the diagonal seems to be patchy or uniform, effectively. And this is exactly what people find in convolutional neural networks. So convolutional neural networks means that we have this kind of invariance as you move along, basically different parts of the network, the weights are similar or change in a similar way. But in convolutional neural networks, the weights are hand-wired to have this kind of profile over space. Here, what we found is that after training the network with real data from this FEF area, we found a structure that resembles visual areas. And you can ask me, okay, fine, CNNs are used for visual hierarchy. Is FEF, why should FEF, which is a frontal area, it's called frontal eye fields, why should it be similar to the visual hierarchy? Because it's frontal and the other is occipital, of course, or sensory. You're absolutely right if somebody asked that. But what happens here is that, remember, the monkey needs to saccade. So we know from other experiments that somewhere in the brain, there needs to be some kind of visual representation of the outside world, some kind of topography, which is similar to visual input and visual maps. And this is exactly where this happens. This happens in the frontal eye field. So we know that the frontal eye fields contain a topographic map, a visual map of the stimulus space. So we would expect this particular area to have similar structure as visual areas. And this is exactly what we find here, this translational invariance, without hand wiring it, without imposing it a priori, as people do in convolutional neural networks. We find it post hoc after training the model using data, basically. So let's move now to slide 13. I won't say too many things because time is flying, basically. So these slides 13 and slide 14 contain some validation of our results of this weight matrix, essentially, against the other methods that people use to find similar stuff. First, here we have a high-dimensional SBD, PCA kind of method that we find correlations between our weight matrix or the latent space in its case and what one could find with SBD. And then slide 14, that I'm passing again very fast, contains another way of validation of our results. Basically, we compare our weight matrix against clustering results based on pairwise correlations. Now you can say, okay, so how can I use your method now, and what does it tell me? So slide 15 now, and I'll be a bit faster from now on. I know time is passing, and maybe we're already. Cecile, feel free to interrupt me when I'm running out of time. So, but I'll come, we're more than halfway there, I would say. So slide 15 now, how can I use that? So first thing is this approach, because like I said, this is similar to principal components analysis. It gives us the intrinsic frequencies, the characteristic frequencies that different neural ensembles oscillate. And this is what you see on slide 15. You have again the 6 possible angles that I remember. 6 possible memories, and then you see different peaks. So you can use that to decode, for example, location. So different neuron ensembles oscillate at different frequencies. So if you had some sort of BCI you want to apply and enhance or suppress the memory associated with horizontal location, for example, or location at I don't know, 240 degrees, this approach tells you where to, at which frequency to stimulate. And similarly, if you go to slide 16 now, what you see here are the components. The previous slide was the temporal axis. Here we have the spatial or connectivity components, the latent states. And the horizontal axis here, you have the different electrodes or location of this multi-electrode array. The vertical axis is the strength, and where you have peaks, basically. These are the dominant populations here, and these peaks correlate with cluster or SVD components. That's what we found earlier. So here it says at which location you can stimulate if you want to suppress or enhance a certain memory associated with a certain angle. And you can see, okay, how is this now relevant for behavior? Of course, you could expect to see results and changes in performance if you stimulate it. I'm very happy if somebody would want to talk more about BCI use and these ideas we can talk later. But we already found some behavioral results. So if you move now to slide 17, what we found is that we use something called characteristic path length, slide 17, from graph theory. And this is basically, if you know this theatrical play called 6 Degrees of Separation, namely that between you and me and anyone else in the world, there are 6 intermediaries. The number 6 is how many steps one needs to take to go from one part of the network, one node, to any other node in this social network. So similar thing here, how many steps we need to take to go from one part of the array to the next one for each ensemble. So we find that the most frugal, let's say, the most economical neuron ensemble is the one that corresponds to horizontal locations. So the brain processes basically memories on the horizontal axis and the vertical axis, presumably more easily. So information propagates faster and it's less energy consuming when memories are on the cardinal axis, on the horizontal and vertical axis. And so this is what we found using basically graph theory and the microstructure, but this is also known behaviorally. So if we move now to slide 18, this is known as the oblique effect. And this was discovered by a physicist, an Austrian physicist, by the name of Ernst Mach, that we all know because of his contribution to aerodynamics and physics, the Mach speed. You see this gentleman up there. But interestingly, he also did psychophysics experiments and found this oblique effect, which is the following, which is that the processing of vertical or horizontal stimuli is better, it's easier for the brain than any oblique stimulus. And this is called the oblique effect. So the brain responds preferentially to horizontal cues. And this is exactly what we found, that the ensembles that represent memories on this axis were more economical and required less energy. And this is exactly the same idea that people use in the graph theory approach with characteristic path length when they talk about energy consumption in large brain networks with fMRI, for example. So this is the equivalent result with these graph theory measures, but at a micro scale. This is the idea we used here. And then let's come to the electric field that we are all waiting for now. I think I have built the whole theory for that, hopefully. So slide 19. So we used basically biophysical modeling and ML and graph theory to talk about the structure and the biophysics in these ensembles. What if now we want to take one step further and see the electromagnetic field produced? And this can be useful, of course, again for brain-computer interfaces and neuromodulation, because besides the targets, we can see what sort of electric field we will need to modulate or basically enhance or suppress. So that's why we want now to have electric field on top. So this is slide 19. And again, we have these new tools from bioelectromagnetism. Let's move to slide 20. So we have this new model for electric field, which goes back to some old work by people like John Ritzel and others in compartmental modeling. What we do here is we consider each neuron as a cylindrical fiber, and you see that on the top left you have different long pyramidal cells with long axons starting from layer 5, 6 up to layers 2, 3 of the cortex. They're elongated, so you assume that of course current moves and propagates along the dendrites. So people have shown that you can approximate that with a cylindrical fiber, which is what you see now on the right hand side here, top row of slide 20. There is a gray cylinder and there is a blue bigger cylinder. And the gray cylinder, what is inside is the intracellular space. And what is between the gray cylinder and the blue cylinder is the extracellular space. So this model is called bidomain model, and it has also been used for evaluating the electric field generated by the heart. And then we put all these cylinders together, many neurons together, and we have, of course, a super blue cylinder, let's say, out of the superposition. And then, of course, a super, let's say, an aggregate gray cylinder. And this is really what we're trying to depict, again, on the top left side of corner of slide 20. These green cylinders basically are similar to what the blue cylinder was on the right-hand side. So anyway, it's the aggregate of the neuron ensemble. We built basically a model now of electric field. How? By knowing what the biophysical neural network is of the neuron ensemble, knowing the connectivity, some of its biophysics, and then using this bidomain model to project neural activity to electric field responses. And this is what you see now, the math, the formula for that is on slide 20, basically. So if you look now at the math here, there is a first equation where it says that the extracellular electric field is the gradient, the negative gradient of the extracellular potential. That's line 1. Now line 2 is that the extracellular potential is now related to the inverse Fourier transform of the membrane depolarization, Vn. And then there is this Wk function, which you see at the bottom row, which basically is expressed in terms of Bessel functions. And this all comes from this bidomain model, where, of course, I have neglected haptic coupling. It gives me an analytical, very easy to compute solution, so it's good for predictions. This is really how I get the electric field from patches of a few millimeters that are occupied by neural ensembles. And the conjecture is that for each memory, I have a certain ensemble, and the electric field of these ensembles for the same memory is the same. That's what that's what this paper is about. And that's what now I want to prove. So let's move to slide 22. Slide 22, top left, you see the electric field on the vertical axis, and then the two axes at the bottom. On the one hand, we have electrodes or locations, and on the other axis is time in milliseconds. So what we have done here is we have simulated sync trial predictions from this biophysical neural network with known validated connectivity, weight matrix, and then we use the bidomain model to project this neural activity from this network to electric field. And then we do that for different trials where the same memory was maintained. And what you see now at the top right is the correlations of electric fields between trials, and you see that the yellow entries here means that the trials are correlated. So I did a large number of trials, over 80%, I believe, are the same, are correlated, which means that the electric field stays the same. And let's move on now to slide 23, where we have these correlation matrices for the electric fields on the left, and then for neural activity on the right. And we see that the percentage of electric fields correlated is higher in the case we use predictions for the fields compared to neural activity, which is the matrix on the right-hand side. So the bottom line here is that electric field seems to be more stable than neural activity. And this is replicated across many electrodes and across many stimuli and memories, and you can find the details in the paper. You see the range of basically electrodes that this happens is between 11 and 27%. And we use a Fisher exact test to compare these binomial distributions of correlations between electric fields across trials and neural activity. The bottom right figure here is the percentage, as you can read, of electrodes with significantly more correlated electric field estimates than neural activity estimates for different angles on the horizontal axis. So what is again the take-home message here? The take-home message is that electric fields seem to be more stable than neural activity. And then on slide 24 is the last result. Maybe I'll wrap up with this, which is slide 24, that it's more reliable. So we have result 1, more stable than neural activity. Result 2, more reliable. What does this mean? It means that it contains more information. And of course, to say that, we need to first to show that it does contain information, which is point 1. So electric field contains unique information on slide 24, associated with working memory, and then point 2, that it has higher decoding accuracy than neural activity. So this is what we saw here in terms of confusion matrices on the top. The left confusion matrix, the true class, predicted class is with electric field. The right-hand side confusion matrix is neural activity. And if you look carefully, you'll see that there is a higher decoding accuracy. And the bottom plots here are basically the accuracy estimates. On the vertical axis, left panel, I guess, is electric field. Right panel is the neural activity. Vertical axis is the electron location. And you have these blue and orange bars. And when there is a blue bar, it means that that is higher than the orange bar. It means that for that particular electrode, the decoding accuracy is significantly higher than chance. And then we did a Werth test, basically comparing now these significantly higher accuracies based on electric fields on the left-hand side plot of the bottom row and neural activity on the right-hand side plot. And we found that indeed decoding accuracy was higher for electric field estimates. So this brings me to the last slide, basically, for this at least first part of the presentation, which is slide 25. Basically, this is what we saw. We saw that we talked about the structure, really, of neural ensembles. We saw that we can estimate the connectivity. We can see how it relates to psychophysics and the oblique effect, and also how it allows us to predict electric fields. If I have 5 minutes, Cecilia will just summarize the remaining 20 slides.
Yeah. Dimitris, don't worry, don't hurry, because the most important thing is that we are able to understand the slides and relate to it. So there's no time limit. So I think it will be more effective if you really took time to make these slides for us. So please explain this at your own time, because we are trying to understand what you're explaining here. So don't worry about the time limit. Thank you.
Oh, okay, I hope I'm not tiring people.
No,
here,
it's
but
a
yeah,
very
thanks.
interesting study, actually. A study that, has a lot of implications in different fields. And I did not notice until I introduced you a while ago about your background in machine learning as well. So I'm very appreciative about the multidisciplinary approach. So please go ahead, and we will ask questions later. Thank you.
Okay, no, thank you so much, and also thanks to everyone, because I just opened this chat functionality here on Clubdeck or Clubhouse, and it says the overall sentiment here, which is very encouraging, and it's green. So as long as it stays green, I can, guys, I can move on to tell you a little bit about and try to test your, your patience here. So let's move on then to a couple of other topics that are very much related with what I talked about. And it shows how this work connects to things that you might be familiar with, like deep neural networks, for example, and I will make some links. So still on slide 25, we talked about structure at the top, the green part of this work. Then I have this, let's say, the red part at the bottom, which is about computation, right? And then, so what these neural ensembles, they do something. Of course, in the simple task we saw, they maintain angles. And that's okay, very, very simple. But what is really the computation they do? And there could be instances where we're not exactly certain about that. We might know how to associate them and how to find their structure with a certain, how to find the structure of an ensemble associated with a certain stimulus. But then in terms of computation, maybe the same stimulus gives rise to different computations. So to test that, we used a different task. I'll come to that and describe it in a minute. But let me read now from slide 25 bottom. So we used a flexible sensorimotor decision-making task. And the key word here is flexible. This means that the same stimulus is processed in a flexible manner, meaning in a different manner, depending on different trials, basically, different context. So here, as you will see in a minute, we had a task where different computations could take place, either categorization or sensory processing. So same stimulus could be perceived as sensory input or could be categorized. And the first categorization is some sort of abstract thinking, which means I take the sensory information, I change it, I manipulate it, and I make it into something else, into a category. So the last point here, I'll talk later about this methodology, about the comparison of brain responses to behavioral models. So let's go to slide 26 now and look at the flexible decision-making task. And again, this is work together with MIT colleagues and the lab of Miller. And this is work also with Marcus Siegel and a science paper in 2015 that they published. So here again, we have a monkey experiment. You see the experiment in sub-panel A. The monkey fixates, as usual in these tasks. Then there is a cue, again, slide 26, panel A. There's a cue for 1 second, and then there is a stimulus for about 3 seconds. The stimulus is, as you see, a cloud of random dots, and there is a response. So the monkey again saccades, either left or right. So why do we have a cue? Why don't we just have the random dots in the cloud? So this is our stimulus. This is what the monkey has to really process, remember, maintain, and base their decision upon, but not only. So now the monkey combines the cloud of random dots, the stimulus, with the cue. So the cue is there, and if you look at panel C, you see different kinds of cues. So it could be for motion, for example, it could be a cross or a star, and for color, it could be a circle or a triangle, panel C. So what does the cue do? The cue says to the monkey, look, if we show you a cross or a star, you should tell us what the motion direction of this cloud of random dots that are moving here from top right to bottom left at somehow at an angle of, I don't know, 220 degrees, for example, if you look. So you should tell us this direction, and you should tell us whether it's upwards motion or downwards motion. So 2 categories, is it up or down? And then if we show you a circle or a triangle, you should tell us the color. And not exactly the color, but whether the color is greenish or pinkish. So basically, depending on the cue, the monkey has to categorize either for motion direction or for color. Now, what sort of stimuli do we show to the monkeys? What are the colors and motion directions? This is what you see in panel B. So panel B here is on the vertical axis has different colors. As I go from top in panel B to bottom, it goes from green, and then it's a bit yellow, as you see, and then it goes down to pink. So the 2 extreme colors are pink and green on the vertical axis, color axis. And on the horizontal axis, you have motion direction. So it could be downwards, if you look at the, on the leftmost edge of the horizontal axis, the motion is downwards. And then it goes a little bit horizontal in the middle of this horizontal axis, and then it becomes, am I looking at it right? Yeah, upwards, I believe. So then the monkey has to tell us, is it an upwards motion of this cloud of random dots that are moving, or is it downwards motion? So is it more green or more pink, and is it more up or more down? And how does the monkey know what to tell us? Based on the cue, like I said. And how does the monkey tell us the category? By saccading, left or right, but the saccade has different meanings in different trials. So basically that's it. So from this slide, please remember what the task is, and also remember panel B. It tells us what the stimulus space is, right? This is panel B now, with the colors and the motion direction. It's like the topography of the stimulus space. So this is the variety of colors I can show to the monkey and the variety of motions. And what does the monkey needs to do? It has to categorize. So it doesn't matter exactly what the color is. It has to tell me, is it more close? Is it a color closer to pink or closer to green? It has to categorize. Now, what we did is we recorded from these 6 areas that you see here, and then we built a deep neural network that mimics this brain hierarchy. Is based on LSTMs. Moving now to slide 27. So slide 27 has a schematic of the network and some details that we don't need to go into too much detail here. But essentially what we did is we built neural networks, deep neural networks, with as many layers as areas that we recorded from that we know the monkey uses to do the task. And then we trained these networks basically to either do the color task, so categorize color or categorize motion. But we didn't only have neural networks that did categorization, we had also neural networks that did sensory processing. So what I mean by that, if now, again, on slide 27, you look at panel B, same thing as before, this is the map of different colors and different motion directions. So you can either train the network to categorize up or down motion and greenish or pinkish color, or you can train the network to classify all possible colors or all possible motion directions. And this I call sensory processing, basically. So because it's not categorization, it's sensory processing. I perceive all different motions without really categorizing and saying as a neural network whether it's up or down. Same thing for color. Why did I do that? I did that because I know that in the brain, some areas do categorization, but some other areas do sensory processing. So it's not that the brain gets straight away the color and immediately, for example, straight away would do says if it's pinkish or greenish or up or down. This happens gradually, as we know. Categories emerge gradually across the visual hierarchy, and there are studies showing that. So slide 28 now is a bit technical. So what we did, basically using this, the data we had and the neural network predictions and the brain responses, is we constructed a representation, the similarity matrices, and an easy way to say what these are is like this panel B that we saw already. So these matrices, these similarity matrices, tell us the topography of a certain space. So what could the space be? The space could be the stimulus space. So there is something called, for example, the behavioral similarity matrix, which is basically panel B, which is the space of different stimuli we can have. So we put all colors and all motions like you construct a map, basically, of how they are arranged to each other at the extreme, for example, edges of this map. On the one hand, you have green, the other hand, you have pink, and in between, you have everything, every other color. Same thing for motion. On the one hand, you have up, the other hand, or the other edge of this matrix or map, you have down, and then all other angles in between. So this behavioral dissimilarity matrix on slide 28 tells us how our stimuli are distributed in this stimulus space. What is the variety? What sort of things does the monkey expect to see? But we can play the same game with the time series that we record from the data, from the brain, basically, or in our data, and the time series we record and predict from the deep neural network. So now instead of colors or motion directions, imagine that you have the corresponding time series. So you have one time series for green and then pink and then all colors in between, and same for possible motion directions. So now you have two more maps, right? You have a map of basically all possible brain responses, and then you have a map of all for color and for motion direction, and then you have a map of neural network predictions for the color task and the motion task, and also when you train the network to do sensory processing, classify all possible directions or colors, and same thing when it does categorization. So you have so many of these maps. Okay, why do I need all this, right? Because here's the trick. We can use now a correlation kind of analysis to compare effectively the brain responses and the time series we get from the brain either to the behavioral maps or to the deep neural network maps. So if we compare the brain responses to behavioral maps, we know what is the domain selectivity, as I call it. So what does the brain area prefer in terms of stimulus? Does it prefer color or motion? Does it prefer category or sensory, or all possible sensory inputs? And same thing by comparing the brain map to the deep neural network map. What is the computation that each brain area prefer? Is it similar, let's say, the time series that I get when I record a brain area? Are they similar to the time series I get when I have a neural network that performs sensory processing, classifies all possible motion directions, or only categories? So this answers the question, basically, what sort of computation does the brain do, and what does it like? So let's move straight ahead, I guess. We'll jump a few slides and move to slide 32, which shows the results of our analysis. So what do I have here? On the top, slide 32. On the top, I have this thing I call computation selectivity. You see the two columns. And then the vertical legend says task. So here you see I have the acronyms of different brain areas. And I split, of course, I have two tasks. Remember, right? The monkey either does motion categorization or color. But then, what is the computation that happens there? It doesn't matter that the monkey does categorization. Maybe some areas compute all possible colors, and some other areas compute the categories. So that's what is this sensory processing categorization. So IT, for example, if you look at the top table, again, slide 32, IT, FEF, and PFC in the motion task do sensory processing, means that the time series of the brain are very, very similar to the time series of the neural network that classified all possible motion directions. But then some other areas, V4, MT, and LIP, the first row, V4, MT, and LIP, for the motion task, do categorization. So their time series are very, very similar to the neural network that basically has categories up or down in the motion task. And the second row here is the same results for the color task. And you can see, okay, you found some correlations, okay, they are significant, you tested for that, you use permutation or whatever, some whatever Bayesian approaches. Can you validate that? So here's the trick. Bottom table here has, the legend is domain selectivity. So here I played the same game, but not with a neural network, but with a behavioral model. Remember, the behavioral model, the behavioral map, is just taking the possible colors and the possible motion directions and put them on a map, creating some topography. And again, looking at correlations between time series of the brain and basically these stimulus maps, let's say, the behavioral models. And same thing, I have the motion, basically task on the vertical axis and color task. And then, of course, I have, again, the behavioral model for sensory classification or categorization. So here, if you look, FEF and PFC now are very similar and do sensory processing in the sensory task, and then categorization, I have it for an MT. The interesting thing now is that there is a consilience, there is an agreement between the two methods, between the correlations I obtained between the brain and the behavioral map, and the correlations I obtained between the brain and the deep neural network map. So it tells me something about the computations that different areas perform. Yes, I think that's the other thing I want to tell you. And I'll wrap up now. Let's move to slide 34.
Can I ask one quick question?
Yep.
You have several pairs of bars here. I'm not sure what's going on the x-axis. So, for example, slide 30I, like the first.
Sorry, your sound is a bit distorted. I don't know. Do you mind typing it in the chat? Maybe that's easier.
Okay.
I can't hear you very clearly. Just here. I don't know why.
Don't mind me. Go ahead.
Yeah, if you don't mind, in the meantime, I can wrap up in the next 5 minutes. I think it's already, or whatever. And then if you type it in the chat, I'll try to answer it.
And
yeah, I don't have time. These are many papers I'm talking about here. So yeah, maybe some things we can come back to discussion. So the last thing I want to tell you, okay, is what if we want to really test these ideas right in humans? That's the last point. That would be, of course, revolutionary if we can do that. I'm not claiming I can do that, but I'm claiming that there is a mathematical way to build an approach for that. And this is the last group of slides, 34 to 40, whatever, 6 that you guys have. So how, for slide 34, how can we test it in humans? The main let's say, difficulty or challenge here is, right, how can I infer this kind of microscopic information that I need, basically, and all this is based on. So these neural networks will have so many parameters because they try to basically capture the microstructure of the neural ensembles at the level of, micrometers, effectively. Can I really reliably infer these parameters using real data? And of course, there's a problem here, but we have a way out using something called statistical decision theory. And the outline of the approach is on slide 36. So what is the situation here? The situation is we will need to use two sorts of models. A neural network kind of model that describes the microstructure, so slide 36. So some microscale invasive model that we can validate using animal data invasively somehow. And then if we want to really capture the low resolution responses afforded by electrophysiology or magnetoencephalography, for example, non-invasive electrophysiology in humans, we need a macro scale model, which will be of lower dimensionality, but at the same time will be efficiently inferred, basically, or the parameters of which can be inferred efficiently. So on slide 36, we have, let's say, the roadmap of how can this be done. We have some papers. There's a communications, Nature Communications Biology paper that describes that, and another paper in Journal of Neuroscience Methods. So the idea is to start with a microscale model with very many parameters, get simulated data out of this, then also have a microscale model that we fit to the same microscale predictions, and this gives us construct validity for the macroscale model that we can then fit to make data. So this use posteriors as priors that you see on slide 36. This is the key idea. So we use something called Bayesian belief updating. So we incorporate microstructure in the macroscale model and then get estimates of macroscale parameters. And this will allow us, if we want to test these ideas, to go from a microscale model of neural ensembles to a macroscale model that can be fit to make data. And we did something similar, not for the deep neural field model I described so far, but for some other model. So if you look at slide 37, so slide 37 is a rat experiment with from Berkeley and a group of young Dan. And here we have a mouse with an optical microscope. The mouse has to do a discrimination task, basically. And what we can do is we record data from all layers of the mouse cortex, essentially. And we have a profile at the microscale, laminar responses, how, for example, layer 5 responses differ from layer 2 responses, etc. Essentially, we have a microscopic, high-resolution data set. This is slide 37. And what we did is we built a compartmental model, as it's called, a model of the sort that Nancy Coppell or Stephanie Jones are using, that describes depth and what happens at the micrometer level. And then using the approach I just summarized very quickly, we basically got some priors for a macro scale model, a midfield model, Wilson Cowan kind of model. This is now slide 39, that basically was able to infer, using here a base factor, whether responses came from superficial or deep layers. And what we did is we had a model, a macro scale model that does not have depth as a parameterization, but at the same time, we chose these parameters to predict the same responses as the microscopic model. So it had microscopic information incorporated, and then it could successfully distinguish, again, slide 39, whether basically superficial neurons in the model really were predicting successfully recordings from superficial electrode tips in a laminar electrode that records from more cortical layers. And this model had more evidence compared to a model where we swapped deep and superficial populations. And this is what you see with the crossed arrows, the yellow and orange arrow, and the base factor response. And now let's go to slide 41, which tells you what is the trick here. The trick uses, like I said, statistical decision theory, and there is a math proof we did. And for you, it might be familiar, for those of you who are familiar with SDT, it's usually used in reinforcement learning and decision making, how we make decisions in the face of uncertainty. There is something called admissible rules, so we make the same error, basically, when we use these rules. The trick here was to reformulate the microscale, the compartmental model, and the macroscale, the mass model, as decision rules that are equivalent. In other words, they have the same errors. What does it mean? We use the model as a Bayesian observer here that wants to infer the true states of the nature, the model parameters. So basically, both models have similar rules, belong to the same equivalence class. So if we prove that, then we can prove that they have the same error, at least mathematically, and then the parameters are equivalent under certain, of course, constraints. So that's the trick. So we could use the same trick described here mathematically to link a microscopic neural network with a macroscopic field model that could be fit in MEC data. And this is what we did in the paradigm on slide 42. So we had human data from MEC responses from different labs. Very simple, slide 42 again, grading stimulus that you see here. We have MEC data, spectral responses that you see from different subjects here. There are vertical axis, it says spectral power, and frequency on the horizontal axis, colored curves are power spectra. You have arrows that denote peaks, and you see the arrows vary between 42 and 50, 50 hertz. So there's some variability in the gamma peaks recorded from different humans. And the question is, what drives this variability? What microscopic properties of the underlying circuitry drive this? That was our question here. So you can see, okay, it's gamma peaks, so maybe superficial neuron activity and GABAergic responses. Okay, but let's see how this is valid. If we want to really focus on whether the variability that you see here is driven by superficial or deep responses, we need to have a model that describes them. We don't have a model if it's a macro scale model because it doesn't have laminar information. The way to access this laminar information is to do the trick I talked about earlier. Use statistical decision theory, construct a microscopic model that has depth, like the one we saw from the rat experiment earlier, then do Bayesian belief updating, have priors in the macro scale model that have this depth information. And this is really the results we found on slide 43. We found for two different data sets with the same paradigm independently that the best predictors of individual variability in this very simple visual perception task in gamma responses were deep inhibitory parameters. This is what is shown on 43 slide. The recurrent connections of deep inhibitory in the neurons or the inhibitory drive to deep pyramidal cells in the London data set. So we had the different MEC data sets. But long story short, we were able to access microscopic depth information using a macro scale model that can be fit efficiently to MEC data. And basically, that's it. Basically, I'll move to slide 46. That sums all this work and the key notions we have discussed. I think it's time for questions after an hour and a half. Yeah, maybe. So, Cecile, do you want to moderate the questions, or shall I read them, or I don't know?
Thank you so much, Dimitris, for the very detailed presentation and for taking time to prepare the slides. So actually, I just want to inform everyone how much we appreciate Dr. Dimitris here, because it took us 3 months just to prepare for this presentation, considering that he's so busy in his lab, and he made a very succinct presentation of the slides today. So I would just like to check with Bisam the sound of Bisam, if we are able to hear it now, because earlier I thought that he was have his voice was so low that he might go near the microphone.
Yeah, and a bit distorted. I couldn't hear him clearly, I believe.
Yeah, so maybe let's go with Sam.
Is this better?
Yeah, it is better, I believe. Yeah.
Okay. I was. We don't need to get stuck in the details. I was wondering about it earlier in the slide there, but I know there's some controversy about pyramidal neurons in particular, whether they're monopoles or dipoles.
So which neurons?
Pyramidal.
Yep.
I noticed with your model a few things, something like around, I think it was slide 20, where you were doing the closed form models. I just noticed a few things about, you mentioned there that you neglected effective coupling. I'm curious what the effect of that would be, and in terms of what would happen if you incorporated that as well.
Very good question. Yes.
and then the dipole monopole thing is relevant in terms of the scaling properties for LFP. So I'm curious whether you modeled these as monopoles or dipoles.
Okay, let's start from the second question. Yeah, I mean, the dipole model we use here is nothing too, let's say, unusual. It's the classical model people use in bioelectromagnetism. Of course, we have considered an aggregate, like I said, not a single neuron, but somehow a neuron ensemble version of this, which one can do. Others, because of the superposition principle, of course, in electromagnetism, even if there is no, even if this assumption of a fiber bundle, of a cylindrical fiber bundle doesn't hold, because of spatial or temporal homogeneities, because this assumes, on the one hand, some alignment over space, and on the other hand, some synchronicity over time, and synchronous firing. One can basically parse space into smaller chunks where this alignment is valid, and then, because again of linear superposition principle, then the aggregate electric field that we computed using this model is basically a bound on what the actual electric field will be, because any misalignment between neurons will reduce the overall electric field, of course, because it will be basically very subtracted. At the same time, the qualitative results, whether, for example, it's stable or not, correlate across trials. This will not change because of the misalignment, because it's all a linear superposition we're computing. I am sure that one could, again, based on principles from electromagnetism, replace the dipole moment with the monopole moment if there are good reasons for that. I mean, again, everybody in the MEG or EG literature uses the dipole model for describing the pyramidal cells and their behavior, but easily one could replace that if they have good reasons with some other model, if they would want to do that. Now, in terms of haptic coupling, I'm looking into that. Actually, that's the topic of the next publication. It's not a huge, of this work, and the next step here, it's not a huge limitation. What we have, when I say we neglect haptic coupling, what one will need to do to incorporate that would be to include a correction term in the neural activity equations. So that basically predicts the effect of the aggregate electric field on synaptic processes and depolarization. So that's all that one has to do. So it is possible to incorporate a haptic coupling within this framework.
Excellent, excellent. I just had one other question and a comment. So I guess I do want to go back to these slides I was going to ask about 29 and 30. I was just confused.
Let me bring them up 1 second if you want to. 29 and 30. Yeah, I didn't have time to really go to the details. Yeah, go ahead.
So these are, I'm just confused about what's going on the x-axis because you have several pairs of bars within each graph. And it seems that each graph is associated with different brain areas. But, like 29 panel I, you have the x-axis labeled with different visual areas, but I'm not sure what's going on in the other graphs. The reason go ahead.
Yeah, first of all, I realize now that my numbering, maybe it's one slide off your number. So it's for me, slide 30 for whatever reason. It says color task. I hope this hasn't been a huge inconvenience for people. So what you see here is the correlations basically between, let me see. This is, correlations between the brain RDM, the brain representation of the similarity matrix, and the neural network representation of the similarity matrices. And this is for the color tasks. So you have 2 bars in each plot. So first of all, we have 6 plots here because we have 6 areas that are implicated in the task and we measure in the brain, and also 6 layers in the deep neural network.
I see layers.
So on the vertical axis, we have 6 layers, L1 to L6, and then we have now for each layer, and then we have the first this empty area. So this tells us the RDM, the matrix for MT, how does it correlate with different RDMs for the network. But of course, remember, for each task, we have 2 networks, one that does sensory processing, classifies all colors, and one that does categorization, so it has only pink or green categories. So these are the blue and red bars, and overall what you see is that for the color task, the blue bars are smaller than the red bars. So for most areas, the brain does sensory processing, so sees all colors. And one can expect that, as I say here, because we have these red-green opponent cells in visual areas already. So already in sensory areas, there is some kind of categorization happening. And this explains also why you can do color decoding, for example, using responses from early visual areas.
Awesome. Yeah, I'm not sure why I read that as A1. So now it makes a lot of sense. I see across the layers. That's actually really interesting. I guess a follow-up question with these monkey studies. I'm curious about how many units were actually engaged by the task and whether that was changing across different brain areas. I'll just leave you with that question. And then the comment I wanted to make is I really thought the end of your presentation was especially interesting. I'm studying cortical columns and dendritic computation. And I think this is really interesting, trying to bridge into things we can measure with MEG or maybe even EEG. So yeah, I just wanted to say that it's super
well, thank you for the questions and comments. I'm glad you enjoyed it. Yeah, regarding your first question, how many units? I believe, for example, in this task, one would need to go back to a paper by Marcus Ziegel and Daryl Miller, Science 2015. If I remember well, usually it's about, I don't remember now, maybe it's about 30 to 40% of the neurons that are engaged, in its trial for different stimuli. Something like that. I don't think it varies a lot between, there are not big differences between stimuli that are carefully selected, so this percentage does not change that much. So there's no bias in terms of one stimulus or the other. We saw that also in the previous task, in the special delayed task, we had these confusion matrices for different angles, and again, if you look carefully there is no bias how well the brain can decode one angle or the other. They're similar. So this means that somehow the information on the paths is similar in the number of neurons, or at least the SNR is similar. That's one. Now, in terms of the drill computations and MEG, yes, that's exactly the line of work we are taking. This, by the way, work that I talked very quickly is motivated by our overall agenda that focuses on testing brain theories and in particular predictive processing. So predictive processing says that the brain basically is a Bayesian predictor or observer, and what the imperative is that the brain does and how we become conscious of a stimulus is by minimizing prediction error. So we have these top-down predictions that presumably exist in deep areas and oscillate at beta activity, at the beta band, and then we have these prediction errors that exist in superficial layers of cortical hierarchies and are in gamma bands. So basically the rat experiment I talked about there is an attention experiment, is an experiment where the rat is stimulated. If you go to slide 37, slide 37 is this optical, basically there's a layer, there's an optogenetic stimulation in the thalamic area, I believe, if I remember well, a laser stimulation in an area called BF, basal forebrain, subcortical area, and this simulates or emulates attention effects. So the drive to the primary visual cortex changes because of optogenetics. So we record activity with laminar electrodes from visual areas, and we emulate the attention effect using optogenetics. And what happens there, what we expect, is to see a change in superficial activity, because that's the known attention effect. And so there is a reason in predictive coding why that's the case, because the precision with which prediction errors are encoded should change. That's the Kalman gain parameter if you do basically Bayesian filtering. And this is what we found as an effect. If you look in slide 40, I didn't talk about that, but there was the main effect was a disinhibition of superficial pyramidal activity. Slide 40, we found that the attention effect indeed changed activity superficially, and we found that using this approach of the microstructure and all this I'm talking about. And the reason we had this question in the first place was because of our desire to test predictive coding that says that indeed attention effects should be superficial. So we need a microstructure model for that, and this is that's what we did. So there is a way, in effect, to link microstructure with MEG, and this is what was the other paradigm I discussed.
Yeah, this is super interesting. One of the things I'm directly testing now is dendritic predictive coding. So predictive coding at the level of dendrites, and our lab is studying layer 1. So we're playing around with these cells that project to the basal forebrain. Yeah, this is really cool. You said something about the number of units not varying across stimuli, but I am still curious if there was any differences between brain areas.
Sure. No, you should look
I'll just look at the papers. Yeah.
It's not in my paper. It's not in 2010 because I work with local field potentials. But in terms of unit, there is a unit analysis in the 2015 science paper Okay. That is referenced in our paper. Yep.
Thank you.
Of course.
I've got a question. I was looking at the oblique slide, I believe, where we're showing like horizontal and vertical, let's say, polarization compared to the diagonal polarizations, and I was reading how it was more effective in a horizontal and vertical ordering, and I was wondering if that maybe has something to do with like photonic clocking in our perception, because I heard Wissam talk about cortex columns, and I was just wondering if maybe there was a correlation between, maybe say our inner ear and how our internal balancing structure and how it affects how that data is stored. It's just real vague, but it just was something that came to mind as I was looking at it. Kind of like when you use multiple polarizing filters, you can block out a lot of the light, or you can reveal it all depending on your orientation. I wonder if there's any studies that went along with that, or maybe it was something on the fly.
Let me remind people which slide you were talking about, and also, so here, what slide was that? Let's see. I believe slide 18, essentially. So what we see here in this looks, like you say, like diffraction patterns. And if you're freezes, that's what comes to mind. What we simply meant here is the orientation of the stimulus, or basically the angle, what is the angle here. So it's not exactly a diffraction pattern. However, I think your comment speaks to the following, for which we don't have any proof, right? But this is just a conjecture I could make. So what the oblique effect tells us is that it's easier to remember, process, recognize locations or anything, any spatial pattern that is along cardinal axis, meaning vertical or horizontal axis. And this is easier to do, more economical, less energy, less metabolic, consumption by the corresponding neurons, compared to any arbitrary angle. And you can say evolutionary, like you were saying, or from a physiological perspective, at an evolutionary time scale, what could the reason for that be? And one assumption one could make is that indeed, evolutionary, from our ancestors for thousands of years that humans exist on this planet, it's much easier to recognize the horizon. People would look at the horizon when they were looking at boats coming or ships arriving at the port. So we much easier recognize horizontal directions and locations, so patterns and vertical ones, than any other oblique pattern. And this shows in experiments, it shows here in our data in terms of the characteristic path length that describes the efficiency of the underlying neural ensemble. That could be an explanation for that. That goes back to our physiology and our evolution at the level now of whole generations and the whole centuries or thousands of years. Thank you. Of course.
Kyle, hi Kyle. Do you have any questions?
I just wanted to just say thank you for taking the time and energy to present to us today. It was fascinating. I guess just for fun, do you want to just kind of riff on what you think, how this relates to regular everyday life for people in your own words, and then maybe elaborate on problem theory a little bit. And then if anyone's done any work in carpentry, I can assure you that horizontal and vertical is a lot easier to process and build than anything with an angle. So I thought I'd mention that because I was thinking about that while I was listening to your presentation. But just whatever you feel like riffing on in how your presentation here affects people in everyday real life.
Sure. No, thank you for the kind remarks. And that's another interesting test or application of this oblique effect. I'm sure, like you said, carpentry or so everyday life. So Mike, for me, and I'd be very interested if people have ideas now or later. You can email me, guys. It's my surname at mit.edu. This is my email address, Pinotsis at mit.edu. So I think BCI is something that could all this work be useful for. And of course AI. I have a slide here that for example, if you go to slide, is it 33? Yeah. So slide 33, we didn't have time to talk about it, but maybe it's a good opportunity now. The bottom of slide 33 says, what do our results mean for AI, right? So first of all, and you can see how everyday this is, I don't know, AI now it's in our, everyday life. So what my take-home message, at least for people who try to develop everyday tools, is that classification of sensory signals is not decision-making, because people tend to conflate, right? I have this driverless car, it needs to classify between, moving pedestrians that are crossing the street and traffic lamps or traffic posts. So classification is not the same thing. That's what we found. One network doing sensory classification, finding all colors, the other network doing neural network, doing categorization, then comparing the predictions of these two networks, doing different things. What the brain does, we saw that different parts of the brain did different things in the same task. Some processing sensory information, classifying all colors, some doing categorization. So, and decision-making was the output of all this process. So it's not exactly the same thing. Classification of sensory signal is not categorization. There are distinct things happening in the brain. We saw that in the data. Now, if we want to build, general AGI, and flexible AI systems, what this tells us also that fixed architectures are not appropriate for transfer learning. What do we mean by that? What we saw earlier is that depending on task, whether it was color or motion, the same brain area and the same stimulus induced different things, different computations that were happening. So if you want to have the same stimulus, but then the brain or a neural network compute either motion or color, then the computations in each part of the network should change, because that's what the brain does. So that's another take-home message for AI people that build these AI systems for everyday use. And the third thing, of course, is AI and BCI, of course. So if we now go back to slides, let's see. '15 and '16, at least on my count, the way I count it. So intrinsic frequencies, and 16 says ensemble locations. So our approach, what it does is it tells you what frequency you can stimulate at if you want to increase or enhance the memory associated with, an angle at 60 degrees or 120 degrees, and then at which locations. This is slide 16. So basically it tells you where and when and how temporally to stimulate. And there are people, actually, I'm organizing a workshop here in London on July 6th. I will send the details to Cecile if I haven't done yet. It's called Brain Data Analysis.wixsite.com. Maybe I can send the link even here. Anyway, we'll discuss some of these ideas. So there are people now using these ideas, or at least I'm suggesting they use these ideas for brain-computer interface, people like Gene Friedman from Johns Hopkins. And maybe let me also copy the link. So you guys, if you want to sign up, you'll be more than welcome. So let's see, how can let's see, okay. So I copied this link to this workshop, and it will be a hybrid workshop. So if you are in London, feel free to pop by. We're in central London. Otherwise, it will be also online over Zoom and maybe over Clubhouse. So the bottom line is people are developing techniques to stimulate neurons, of course, as you know. And then you have a patch and you have a neural ensemble. And what people do so far is try to, by trial and error approach, stimulate here or there at a certain frequency. What we have here is a principled way, an organized, systematic way of saying where you should stimulate and at which frequency if you want to suppress, for example, a certain memory associated with a certain location. We haven't done that, but we could do that. And yeah, that's the other. It's not so every day, but it could have real-world implications. In paraplegics, for example, the sort of work that you guys have discussed with Miguel Nicolelis, I understand, in previous sessions, etc.
So,
thank you very much. Just quickly, if I've got a friend there and want them to meet up with you at that conference, would that be acceptable? Just have them send an email to you?
Yes, please send me, like I said, an email. I'm happy to meet outside or inside or outside the conference. My email is my surname at mit.edu.
Wonderful, thank you.
Of course.
So, what I'm kind of hearing, and this is probably naive, and most definitely is, but I'm hearing like, okay, so there's a vertical and horizontal structure of a thought, and Kyle, that made a lot of sense when you're saying it's easier to build vertical and horizontal than it is to put a pitch on a roof. But it's had me thinking kind of like I've been thinking about rise and run, but I've also been thinking about rise and run as it relates to thought propagation, as far as like it sounds like I'm getting an echo. But it sounds like you're using an evaluation hierarchy, kind of like a gambling with cards or something like that for rise and run. And what about like a binary? What's the binary circuit or wave propagation that promotes or suppresses? That's the depth of thought. Is so you have rise and run, but the gates that say yes and no, as assessment occurs, I'm not really understanding how you're evaluating for the
Sorry, when you say rising run, what do you have in mind?
Well, you're talking about, y'all are saying that the hierarchy. So like rise and run, to calculate the angle of a roof slope, you need to know, like generally speaking, here in the United States, we use empirical. So say I want a 45-degree angle on my roof. Well, the rise and run's at 1212. But if I want like a 22.5-degree, the rise and runs, I think it's a 512, 5.5, 12, and so that gives you the angle calculation. So that may be a vector of that you're assessing towards a point internally. The rise and run allows you to kind of create a vector or a thought flow, an orientation towards where your thoughts may want to go inside, but because this hierarchy is row and column based, then it's kind of working like a voxel or like a point cloud. And then Rise and Run's like, hey, I have an idea that looks like my slope or my pitch or whatever. That looks like the slope of idea that I need to go. And as I go through, what's the binary, have you investigated the binary logic gates that go yes and no, keep going, because that'd be into the decision making where it's like, oh, that color's wrong, so now I have to change my slope and my slope of intention, or I have to create a vector around this.
So what I think might speak to your to this line of thought, so I think what you're first of all, the tasks we talked about, right? They are much simpler than what you are describing. So it was a special delayed saccade task, or this flexible sensorimotor decision making with the rules and the cues and the context for color and motion. However, if you go back to our initial slides here, where we talked about oscillations, and for example, go into slide, what was that? Yeah, for example, 6, and you read slide 6, where I talk about oscillations and different domains of application, and there's a point here, 1, 2, 3, 4 from the top, about binding theory in Wolf Singer's work that I alluded to, binding of input to cortical representation says. So what happens there is basically maybe what you're describing is how do we internalize and create cortical representations of various dimensions associated with the stimuli, and maybe there's some kind of also internal processing and transformation of these stimuli, some binding together. So we expect this also to occur through some kind of synchronous activity of different neurons. Let's say I have a part of the cortex that represents fruits, and I have a part that is preferring apples, and then I have a red apple and a green apple, and I show the image. There should be some kind of binding process mediated by oscillatory synchronous activity that creates a representation of green apple versus red apple. And this happens, of course, internally and transforms different stimulus dimensions into a coherent distributed representation. And so, in principle, one could think of paradigms where this happens, and actually, like I said, people have used this temporal binding theory and these ideas to test basically how these representations are formed. However, we don't have so far good, let's say, because all these measurements in humans, of course, that perform these tasks, all the measurements come from outside the head. We don't have a way of directly accessing what's going on inside the head and at the level of these neurons that maintain thoughts. And in a sense, our work speaks to that. The other thing is this electric field angle that we put, that we brought to the table could be a way of achieving this kind of internal transformation of thoughts and this temporal binding. And there are people who have suggested that electric fields mediate cortical representations and even consciousness, if you will. The field property is useful because it allows this binding to happen at the speed of light, essentially different parts of the brain to communicate very, very fast, because that's the level at which field commutations would emerge. So let's say I have the image of an apple, and then I know it's red. So different parts of the brain, like I said, respond to apple and red or any color, and then neurons synchronize to construct a coherent representation of a red apple, and this could happen very fast using electric fields locally inside the brain, and this is better. I mean, if we do that over fields, of course, this can explain how this happens so fast compared to neural computations, a synaptic processing, which requires some delays because signal needs to propagate from the apple area through synapses and from one neuron downstream neurons to the other neuron to really achieve this binding, while the field aspect that we're talking about here could allow this to happen instantaneously at the speed of light, like I said. So that's, of course, that's a conjecture to be tested or not. I don't know if we can test it really.
Well, it's kind of like, part of me's thinking about wave propagation and like, also, the, electromagnetic rotation of photons, right? And it sounds to me like the brain structures, they will accept kind of wave propagation that's clocked accurately to land in these horizontal rows and vertical columns, but it's less efficient if that wave propagation is not landing in that clocking. So
that's the assumption. Yeah, you're absolutely
so it alludes to a superstructure of like clocked quantum reflections or whatever, wave propagation is almost like what I'm visualizing is like if you sit there and put a restriction on your thoughts, whatever it is, I don't want this out, I say I don't want this to happen in your thoughts. Once you shape that then by putting that I don't want, you may be like doing that polarization termination of photonic cells by maybe clocking that output at a 45 degree, which will or at an opposing polaric field, which will stop the propagation of that non-desirous outcome. So like putting some Venta black for where the in your brain for no, I don't want to go that direction or something like that. Or this is interesting to me, but I don't know if anyone makes sense of what I'm trying to describe because I'm just visualizing it.
Just wanted to see if Ethan had a question.
Unfortunately, I didn't get to hear most of the lecture at all, but so this may be wildly off topic and having to do with something we're hoping to talk about soon in other rooms, but I'm just curious, and your answer might be no, and it's a very quick response, and that's it, and that's fine. I'm just wondering what, if any, the ideas of Gerald Edelman have on you, guiding your thinking and your experiments, and if they do, just some general comments about that.
Sorry, what was the name you mentioned?
Gerald Edelman, a lot of his theories.
I'm
just wondering if it informs any of your research or guided your thinking, if not, that's a fast answer. If so, then maybe just expand.
I've read the classical books, of course, Edelman's books as a general guide. I mean, there is, I think, Giulio Tononi who has worked with Edelman, has this phi theory for consciousness, which I've been thinking a little bit about. Although I don't work on consciousness, and I'm not definitely an expert on that. But yeah, that's the closest, at least this work, I would say, goes or gets the closest touching point or point of contact through, the phi theory. The point is that at least this theory doesn't talk a lot about the biological implementation of memories or conscious awareness, which is the focus here. Here is, like I started, the focus is on structure, on biophysics, on the energy and the energy field, for example, of neural ensembles. So that's our focus here. I'm not sure which other part of Edelman's. I'm sure Edelman has done many things that I'm not aware of. So I don't know which one you might be alluding to.
No, I was just getting a sense. That wasn't really anything in particular. Just fishing. Thank you very much.
What was this theory of consciousness?
It's called the phi theory. By I think it was a student of Giulio Tononi.
It's less him, I think. I think he came on and interpreted some of the results. I'm getting a sense later and used it to add to it. But I think
incidentally, Dimitris,
can you send me this?
Incidentally, I would like to say that, we have over the weekend related and upcoming rooms, which might be of interest to you, but I think it may be your 4 a.m. So it will be during our usual club time, because on Friday evening we will have a room about a research on consciousness at the point of view of physics. There's someone who will talk about consciousness on the relativistic point of view. And then on the night after, over the weekend, on a Saturday evening, we will have some research conducted by Singaporean and Japanese researchers. They are trying to use brain-computer interface with optogenetics and metamaterials. And yeah, I think it's very interesting, but it may be 4 a.m. with you. So I would also like to make this announcement to our audience that in a way our rooms here are becoming more and more connected. So yeah.
Yeah, what I would recommend, thanks for sharing that, and I'm not sure I'm able to follow at 4 a.m., but I would definitely listen to the recording afterwards, and just brainstorming with you. I know you're very kind and open to. It might be nice, besides this, let's say, presentations or these talks, to also have, since you have now a network of people that you have created, in a sense, that work on related questions, to even at some point organize a round table at a convenient time that everybody might, three speakers, for example, might prefer, and then we discuss a little bit. So we could talk, instead of me giving you, a 2-hour long or an hour and a half long summary of my work, I talk about, for 15 minutes, what are my interests, and then other people could talk about theirs, and then we discuss, open questions or try to find how we can all address challenging and questions or open questions in the field. And I think that would be also a very useful contribution of your clubhouse room here.
Yeah, that would be very great, because I'm just thinking about a panel, and earlier, before we had this room, when we had the call, so many things came into my mind as related to where your study will take, will encompass the researchers of those who are making parallel works on that. So yeah, we shall do that very soon. So, I would like to take the opportunity. I have tried to request Serge to come up, and I think he raised his hand earlier, because I see that he has questions here at the chat, and I would like to refer to the chat to give an opportunity for those who are asking questions, for their questions to be read. But Serge, if you would like to come up, we are certainly inviting you. Please raise your hand. So initially, the first question of Serge, which came, I think, just after you started the talk, Dimitris, is how do you set up your electrodes matrix? Specifically, how can you synchronize or make sure, say, if you have two monkeys, the matrix located in the same area of its monkey brains? So
yeah, that's I can read the question now. I think for people, it's maybe if you scroll up to the chat and the very top, and then you scroll a little bit down. There's a long question by Serge. How do you set up your electrodes matrix? Yeah, so you mean the array. There are anatomical landmarks that we use. I don't do the surgery myself. It's a very complicated process. Monkeys first undergo fMRI scans of the brains to find exactly, because of course each brain is different, right? They don't have the same brains like humans, don't have the same brains. And you can say, okay, how do I know that I'm recording from the same area if I'm gonna basically use claim that I can replicate my result into different monkeys. How can I claim that? I need to record from the same area. How do I know that? Because since the brain itself is different, I'm looking for, again, landmarks. Those like the sulci, if you remember my slide, if you go to slide, which slide is that? Slide 11, for example, you see monkey C and monkey J, and then this PS line on a schematic, top left, so slide 11, the special delayed response task. And we try to place the arrays, these electrode matrices, at the same anatomical location as defined by these landmarks. But of course, that's not the end of the story, because maybe anatomically is the same, but what about the functional information there? Does the area do more or less the same? So then there is a technique called receptive field mapping. So then you show stimuli to the monkey that you know this area prefers, is selected for. And then you measure responses and you really make sure that the area in both monkeys is anatomically the same and functionally the same, namely it responds to the same stimuli in both monkeys. And that's how we ensure that really it's the same location, basically, both using anatomy and function. Got clear? Hopefully that answers the question.
Yeah, I think those are the only questions that we see so far here in the chat box. Yeah. So, Dimitris, I would just like to ask you a question for the sake of resetting the room for those who just came in. So, you had spoken about representational drifts, and so for the sake of resetting the room, I would like to ask you, how do these electric fields try to unify as a conductor the representational drifts that we have?
Absolutely, yeah, that's great. Thanks so much. I don't think I had time to emphasize everything in the detail I should. That's one point. So I'll go back then, and also for people who didn't, were not here maybe since the beginning, I'll point first to the slide that talks about this. So please look at slides 22 and 23 that show you the electric field. So first of all, what is representational drift, right? Representational drift is this experimental observation that the neurons that are selected and are maintaining a certain memory, like a certain angle, 0 degrees, 120 degrees, whatever, horizontal location, vertical, or any other oblique location. So the neurons, in each trial that I show to the monkey the same angle, and I ask the monkey to remember the same angle, the neurons that maintain the angle are different. So they drift over time. So the representation drifts, and the name is representational drift that we give to this phenomenon. So people don't know why this is the case. It's a mystery. And there are several explanations that relate to how learning happens and plasticity happens in the brain and how memories are formed. My suggestion here is that this is absolutely fine, and this speaks to something that is known in the electromagnetism as the non-uniqueness of the electromagnetic inverse problem, non-uniqueness. So non-uniqueness means that the same electric field or electromagnetic field can be generated by an infinite number of different neural sources. And we know that to occur in nature. So electromagnetism is the theory that describes how basically charges and currents occur in nature. And we have that, in electricity in our houses. We have that on the sun, there are charged particles. The aurora borealis, for example, is something like that comes, charged particles that come from the sun to the atmosphere and the earth, and they produce this very nice, very fascinating, green lights that we see in Iceland and Norway. Anyway, so there are charged particles everywhere in the universe on Earth, and electromagnetism describes how these particles move and evolve over time. And they say that they obey, basically their motion and evolution obeys the fields that are generated, similarly to how we obey the gravitational field. So we have people on the Earth's surface and we move and we have a certain weight, which means there is some gravitational force that we are subjected to. And if you remember, Elon Musk and all these recent, orbital experiments putting people into conditions of weightlessness, this is what happened. There is no gravitational force there. So, but we are all subjected. We don't understand that, of course, because we are born into it. But if we go up to the edge of the atmosphere and the edge of the gravitational field, we will understand that, of course, that there is something like that. Anyway, the field generates basically, is the output basically of neural activity and it's stable. That's what now 22 and 23 shows, that there is some stability. So the same memory is associated with the same electric field across trials, and maybe there is some variability in the neurons that generate this field and this memory. And this variability is what we call representational drift. So the neurons change, but the field doesn't change. And this is fine because electromagnetism says that that should be the case. There are multiple ways, again, to generate the same field from different people, from different sources, like different people on the Earth. We move around, but the field generated is the same, essentially.
Yeah, thank you so much.
Is it clear? I don't know.
As I tried to.
So yeah, that's the correlations that we saw on 22 and 23. The yellow matrix says that the trials across trials matrix on slide 22 and also 23, yellow means that the field is the same between different trials. The two trials are correlated. And then slide 23 shows two yellow matrices, left and right. The left is the electric field, the right is the neural, the correlations of neural activity. Of course, you said there is also correlation in neural activity, because remember here, neural activity is stationary, its memory doesn't change much. But what is important? The important is that the correlations are more, we have more correlations when we look at electric field correlations compared to neural activity. So it's more stable than neural activity. The reason neural activity is stable and correlated is because we have memory, so we have stationarity over time, not much changes. But of course, there is a little bit of change of the neurons, so the correlations are not as good as when we consider electric field. And this is what the bottom plots on slide 23 shows. It shows the percentage of electrodes where the correlation between electric field estimates across trials is higher, and we have more correlations when we look at the electric field estimates compared to neural activity estimates. And for all electrodes, and for all, or for a large number of electrodes, and for all stimuli, it's always the electric field that wins. So it's always the electric field that is more stable compared to neural activity.
Yeah, thank you so much. Now I have more questions in my head than before, but maybe that's for another time because, coincidentally, preceding to this, we had been making maybe for a month and a half so many discussions about, the electromagnetic field, and in fact, few weeks ago, we talked about Faraday and Maxwell, and we tried to dive deeper. We also talked about Tesla, and we tried to dive deeper into optics, vibrations, and electromagnetic fields. And I was just wondering about the implications of your study as to the smallest and to the biggest thing that is being affected by that. And also in relation to what I mentioned before, the study of Michael Levine about electrophysiology, which is very, very interesting, and it's very much related to you and how you will be able to try to collaborate on doing future studies on this, because this is very much unconquered field and there's so much to learn. So I will just skip the questions in my mind now because I think it's a discussion of several days. So, but I find your study really, when I saw it, I was really amazed because it encompasses a lot of disciplines and it has a lot of implications. Maybe the question that I can ask for you now is just, having found this out, what is the next level that you're taking it to in your lab?
Yeah, let me make a quick comment about Michael's work that you alluded to, and then I'll tell you about also the next step. We have a few more minutes, I guess, but I will be very quick. So one thing is, so this morphogenesis story, one could think that evolution has somehow, or the organisms that survived have a certain configuration of electric fields that are optimal somehow. Again, this is just speculation. So evolution, one way to measure fitness, evolutionary fitness, is by electric fields. So when we have the genesis of an organism, right, when the story of orthogenesis, it means that the parts of the organisms, the configurations, are put together in such a way to have some optimal electric field. That would be a point of contact between this work and Michael's work, I believe, that we could describe what these electric fields are generated by neural ensembles, and then, of course, Michael's work would show how this is really achieved. So how do we get that out? And in a sense, all this work by Kohonen and the Kohonen maps and self-organization, which underlies also what we did here, speaks to that. Like, how do we get a bunch of neurons that are brought together and form a circuit, a cortical circuit that can efficiently process information? So we start, you can think like the red and green opponent cells, right? Very simple categorization of colors in our elementary visual cortex. To do a computation like that efficiently, we need to have some wiring. How do we get this wiring? Through evolution. What is the rule of evolution? There's some plasticity rule, some evolutionary rule that leads to an optimal circuit. What defines optimality? The electric field in the very end. So we could think, and actually I believe there is some work done with genetic algorithms that took a bunch of neurons and units, evolved them over time, over generations, and the goal was to have an optimal electric field. So we put them together, neurons have currents, they produce fields, and then they need to somehow get the most economical field. So that's the point of contact between this work, the electric field work, and the way we have achieved and have made predictions of this field and this self-organization literature. Next steps. One next step would be exactly that. Second next step would be the BCI work that I alluded to in neuromodulation, finding targets, which neurons to target and at which frequencies to suppress or enhance certain memories. That's second direction. Third direction that I've already a little bit spoken about is really looking at the faptic coupling. What does it mean? How can this be achieved? Is it there or not? In which paradigms, which brain areas? Because we know that the faptic coupling there is evidence for that, experimental evidence, but usually this is in vitro, meaning we have a brain from a rat, we cut it into slices, then we put two slices, let's say neighboring parts of the brain, to get next to each other, we stimulate one, we see how this stimulation affects the activity of the other neighboring slice, but this is not in a living brain, this is all done in slices, in vitro, and people have found evidence for interaction between one slice and the neighboring slice. So basically, if you measure now from the neighboring slice, let's say, the activity there changes if there is another slice next to it, but this is not a living brain. So how can we really see whether there is a faptic coupling in the living brain? That's another direction. And there is a social way analog to that, which is we discussed in an MIT news piece. It's when you have, for example, an orchestra, right? Think of an orchestra. Now, its neuron is a person, a member of an orchestra that plays a song or something, a symphony. So if you talk to people who are members of an orchestra, musicians, you will see that for the same musical piece, for the same symphony, they are affected by basically the conductor. So the conductor that differs is different, right? The director of the orchestra. We discussed in an MIT news piece. It's when you have, for example, an orchestra, right? Think of an orchestra. Now, its neuron is a person, a member of an orchestra that plays a song or something, a symphony. So if you talk to people who are members of an orchestra, musicians, you will see that for the same musical piece, for the same symphony, they are affected by basically the conductor. So the conductor that differs is different, right? The director of the orchestra. But then the piece is the same, the song is the same with the symphony, and then basically the way people play their music, their instruments, changes depending on the conductor. So this is some sort of basically top-down influence, some sort of a faptic coupling that presumably in an orchestra takes place and probably also in the brain. So that's a very exciting direction also we're looking at.
Are you familiar with Wade Rager's work at Harvard?
Sorry, which one?
Wade Rager.
Can you write it? No. Yeah. Do you mind writing it in the chat?
I'll write it, yeah. They did really interesting stuff recently on effective coupling in the cerebellum.
Great, yeah. No, yes, please. Yeah, that would be very, very useful. Yeah.
Yeah, I also would like to hear Wissam tell us about it later. Yeah, in the meantime, we have Ali here who came up.
Hi, glad to be here.
Hello.
Ali, do you have some questions or comments?
Yeah, sir. Yeah, sure. I want to ask Professor Dimitris that if we can mathematize and model altered states of consciousness in the terms of mathematical neuroscience, and then model them into AI. Thank you.
Ooh. Yeah. Consciousness, yes, I alluded to that, and there's definitely some, it's obvious that there is a link. I mean, there is a bunch of theories, right? Electromagnetic theories of consciousness by people like Susan Pocket and Joe McFadden and other people who say that, instantiation of consciousness can be found in electric fields of the brain. So in a sense, the model we have here for these electric fields of memories, in a sense, is somehow a very simple example of a mathematical model of consciousness, perhaps, or perhaps not. I wouldn't claim that necessarily, but at least shows us a way of achieving that kind of thing. There are not that many models. Like neuroscientists are a bit careful sometimes when talking about consciousness because we don't need them, let's say, for many things, right? So the focus is understanding computations in the brain, understanding mental health, understanding what goes wrong, for example, in psychiatric disorders. There's not much space for consciousness, and the other question is how do you really define consciousness? We all know it's there, but there is no absence of consciousness easily. Of course, there are studies on coma patients or anesthesia studies that one could take as proxies of absence of consciousness. But there are not that much. Like I said, phi theory by Tononi, for example, and Edelman, I believe each one of things is one of these mathematical frameworks. Another is the global workspace theory that speaks to similar questions. There are these, again, bunch of theories, not so mathematical, the electromagnetic theories of consciousness. Maybe you guys have heard something else. Yes, I think going back to your question, I think that it could be tested at some point. And in a sense, my take would be to really focus on basically paradigms like the one we discussed here with perceptual decision making, memory, and try to see basically what is the fields, what kind of fields these conscious thoughts or memories generate. And there is an evolutionary component, of course, to that somehow neural circuits that can efficiently, and biological circuits that can efficiently perform tasks should lead to electric fields or electric field configurations that have some intrinsic optimality, and this is associated with this conscious thinking and efficiency in the task compared to, let's say, a person who is disabled or has some disorder and cannot perform the task. Then the electric field should be different and suboptimal, whatever that might mean. And this can have connections again to BCIs, right? Because in a sense, what brain-computer interfaces we try to do is restore this kind of healthy, optimal electric field compared to the pathological one, this configuration that is suboptimal and cannot do the task. And then again, if you have an AI now, one can say that AI will never be conscious because it does not do computations based on electric fields. But if one believes that electric field computations have something to do with consciousness, then the only thing we need to do is build AI that does computations based on electromagnetic fields, and this will be more efficient, and this will endow AI with some kind of consciousness. And there are people, like I said, like John McFadden, who have promoted this kind of thinking.
Would you mind sharing the names?
Thank you so much. I just want to please suggest me some paper in that matter for the last part of your answering my question. Yeah, thank you.
Of course.
Sorry, I was going to ask if you wouldn't mind sharing the names you brought up in the chat.
I can share the wiki page that gives an overview of this.
Oh, perfect.
I think that's these are called electromagnetic theories of consciousness, really.
The other name that comes to mind is Miguel Nicolelis. He also has a EMF theory of consciousness.
Okay, great. I should have, then it might be in this, okay, so let me copy that. For those of you who are excited by that, everybody's excited, of course. Let's see. Right, is this? Yeah, it should work. Yeah.
Thank you.
With some, since you shared some, the name here on the chat of Wade Rigger, can you please tell us? I'm just curious, what is his work about for the benefit of all of us?
He works in the cerebellum, but he's just he's a unique neuroscientist. He's very, very rigorous, but they've done a series of papers. One recently, as recent as last year, I think, where they talked about the climbing fiber synapses. I won't get us off topic on cerebellar circuits here, but they are demonstrating effective coupling there, and they use a mixture of in vivo and in vitro recording for that paper. I can drop that paper I'm talking about specifically. I can drop that in the chat if you'd like.
Thank you. I was actually reading a bit, but we'd like to hear about that later. So. And Ali, you were saying?
I told you. I told Professor Dimitris that if it's possible, mention some papers and outgoing works in order to to mathematize and make some mathematical models of altered states of consciousness, and also to model them and use AI as those conscious agents, if that's the case, of course. So please share some links. Thank you.
There is a recent research of interest in psychedelics research on the one hand. I know UCSF, for example, has a Center for Psychedelics. There are many people working on that. I guess again, some Wikipedia page will have a wide variety introductory coverage of this research. The other thing is, there is more recent work also by colleagues at MIT that I'm aware, and others, and Harvard. Gabriel, for example, if you know Gabriel Kremen, or typical, they train neural networks to access basically or alter configurations, in the sense that they don't train them on natural images, but they show that they can have responses that are outside the range of common responses one could get if you train, when they train a network to recognize natural images, for example. So that could be another angle to the same question, altered, accessing representations outside what evolution might have come up with by some kind of reverse engineering, is similar in the sense to these deep green Google images that you guys might have seen, which look a little bit like psychedelic images. So that would be a couple of pointers from my perspective for that. Psychedelic research.
I just took Professor Kryman's class, and he was on my PQE. I had to get by him for my qualifying exam.
Yeah, did you talk about that?
Psychedelics?
No, his neural network work with these novel representations.
Yeah, it's really interesting. I think you're talking about where they had the electrode in feedback with the genetic algorithm.
Yeah,
good stuff. So I dropped that paper in the chat.
Yeah, please. Yeah, I think maybe Ali wants to have a look.
Thank you so much, both of you.
Of course. Yeah. Maybe we'll run out of questions, I don't know. Or getting tired.
Yeah, we are trying to copy the links in the chat, so people are a bit quiet. So I just don't know if there are more people who would like to ask questions. Please raise your hand. And I know that Dr. Demitris has been here for 2.5 hours now, more than 2.5 hours actually, right? Yeah. So I want to ask if how much more time can you share with us, Dr. Dimitris?
Yeah, I mean, I guess we'll wrap up not so long from now. I understand that that's your common also. It's a usual duration of this, and I'll have to go. Obviously, it's getting here past my past dinner time, so maybe at some point.
Yeah, but anyway, we can always invite you back here, because I think if we look at your study, there might be many questions that we would like to ask again. And I know that your lab is making a lot of papers, making a lot of studies, so we would like to invite you again.
Of course, it would be a pleasure. Join back, and like I said, if you organize also a panel, I'll be more than happy to come back.
Thank you. Please do come back. Greatly enjoyed it. Already contacted somebody in London to see if they can attend that conference and send you an email. So if you see an email saying Kyle sent you,
there is a form. Yeah, there is a registration form. If you go to this brain data analysis website.com/main, there is a registration form one can use. Hopefully it works, because we had someone saying it didn't work, but hopefully it works. Or if not, please let us know. There is an email address also. You can email.
We have yet to talk about streaming some part of the conversations there on July. Streaming it here in.
That would be great. Yeah, if you can do that.
Yeah.
I would really appreciate that.
So yeah, we really look forward to that. And so yeah, I think we will Wissam, do you have other comments? We will try to prepare to wrap up the room soon.
Yeah.
No other comments. This is really interesting stuff. This is stuff I think about a lot. So I really appreciate the presentation. Perhaps I can follow up with you with some emails if that's all right with you.
Absolutely, yeah. Feel free to contact me anytime.
Sounds great. Thanks for the presentation, and thanks for everyone that jumped up and asked questions and participated.
Yeah, and also thanks for coming with some, because I know you're working today, so really appreciate it. So with that,
really appreciate your work, and thank you, Cecile, for the good club. I just wanted to show my appreciation.
Thanks so much too. Thank you for your support. So, I would just like to announce the room that we will have tomorrow. Someone is going to speak about the alternative theory of gravity, MOND. So it's a prerequisite to a room that they will have about their paper on Sunday morning, actually. So please come. And anyway, we would like to thank Dr. Dimitris Pinotsis for coming here and for the generosity of his time and knowledge, and for accepting our future invitation already as of now. So,
Cecile. Thank you, Cecile, and everyone for their attention. It was a pleasure, spending time with you guys. And anybody who has some question or some idea they want to discuss, especially along BCI, memories, neuromodulation, or whatever, haptic coupling, and all these ideas we talked about, electric fields, feel free to contact me. Like I said, I'll write my email actually in the chat, and I also send a link to the conference. So my email is my surname, pinotsis@mit.edu, and I just send it in the chat.
Dimitris, probably we can do another room on just Q&A. So about the many about several topics related to what you do. So let's see.
Anyway. Let me know. Yeah. You have my email also.
So we'll talk about it at the later. So we have a tradition here. Every time we have a room in quantum photonics, there is Darcel who played music earlier to open a room. And we would like to invite him to close the room, but I'm not sure if Darcel is up here because I cannot see him in my app. Sometimes my app is acting up. So are you here, Darcel? Or not yet? I will.
I see him in the audience.
Can we please bring him up with some? Maybe Darcel is raising his hand. I will just exit a bit first and come back because my app is freezing.
Yeah, I'm trying. It's not really working out. Okay, that worked.
I'm here. Thank you, Wisdom. Cecile, should I go ahead and start, or do you have more announcements?
So now I see Darcel up here. So, Darcel, can you speak?
I can. I am here and ready for liftoff. Thank you, Cecile.
Okay. So, with that, I would like to thank again our very special guest, Dr. Dimitris Pinotsis, and all the panel here on stage, and those of you who are in the audience. Thank you so much for your support to Quantum Photonics. Please follow Quantum Photonics if you're not following it yet. We will be having more discussions and more interesting rooms. And with that, I would like to pass the mic to Darcel to help us close the room.
Thank you. Thank you, Cecile, and thank you, Doctor. I really enjoyed it. As always, I learned a lot. We're going to start our countdown with lucky number 5 to close the room. Is everybody ready?
Ready.
5.
5.
5.
4.
4.
33 and a third. Dos seconds. Doctor, you are number one. Go ahead and call it out.
One.
Yes.
Thank you so much, Dimitris. See you soon here in Quantum Photonics. Yeah. So thank you,
Thanks,
everyone.